So, 70 years ago, Aldous Huxley, anticipating the transformation of human life through advances in biology as the final and most searching revolution, asserted this really revolutionary revolution is to be achieved not in the external world, but in the souls and flesh of human beings. In the decades since the first publication of Brave New World, amid the accelerating pace of discovery in genetics, developmental biology, and the laboratory production of life, there has been increasing appreciation of Huxley's prescient concerns. Yet throughout this period, limitations in our tools and techniques for specific and efficient modifications to genomes have been a major constraining factor for advances in biotechnology. Now, however, in what MIT Tech Review has called, as you heard, the biggest biotech discovery of the century, CRISPR-Cas9 and related techniques promise easy to do, inexpensive, and highly precise genetic deletions, insertions, and functional manipulations of genomic processes across the full spectrum of living beings. This holds great promise for advances in agriculture and animal studies and basic biomedical research and therapy, but opens fundamental questions about our role within the natural order and the use of these technologies in shaping the human future. CRISPR-Cas9, it's an acronym. It stands for some technical thing about the molecules. CRISPR-Cas9, which is adapted from an immune system deployed by bacteria to protect themselves against viruses. It's adapted now to be used as a tool. It has been described as a genetic scalpel or as molecular scissors, but it's more rightly compared to a Swiss Army knife. It's composed of two basic elements. There's a guide RNA that locates the place on the DNA that you want to make an operation of some kind. And then there's scissors, but the scissors can be modified to do a range of different functions. As scientists continue to explore CRISPR's system in the many natural variations, and then selectively, scientists are now selectively engineering clever new modifications in its targeting and operations, they're developing a whole new genetic toolkit with functional applications at every level of genomic process. The breadth and flexibility and precision of these new tools it's absolutely breathtaking, and it's opening a vast increase in the range and complexity of experimental possibilities, theoretical insights, and practical applications. For example, individual genes can be strategically disrupted or selectively altered. Sorry, that's kind of a low pixel photo, and some of them will be, but I had to take something from the web, Anyway, individual genes can be strategically disrupted or selectively altered to the precision of a single base pair. Remember from high school, the sort of rungs of the ladder. It's that precise. And with that, you can study the role of these genes or their specific forms in healthy development and disease. New genes or regulatory elements that control the genes from different species can be inserted into agricultural crops to provide drought resistance or more rapid growth or enhanced nutritional value. Amazing, wonderful possibilities here. Transcription, you remember from high school biology, the DNA is transcribed into the RNA, which is then translated into proteins. Transcription, the first step in gene expression, can be selectively blocked or promoted, or placed under inducible temporal and spatial expression subject to light or chemical signals, allowing study of tissue differentiation and organ development. Likewise, whole patterns of epigenetic effect. Epigenetic means outside influences that impose on genetic function. So whole patterns of epigenetic effects on gene operations can be simultaneously studied, delivering new insights into environmental impacts or greater understanding of pathological processes in neurodegenerative diseases, cancer, and a range of things. Moreover, recent discoveries of CRISPR mechanisms that directly target RNA molecules extend the range of interventions beyond transcription all the way to the control of the quantity and chronology of protein production, and thereby to refinements of cell organization, operations, and functions. If you can control the proteins in life systems, you can have vast control over those systems, and that's what we're gaining. Together with deepening knowledge of genomics and our exponential increase in gene sequencing capacities, these new tools for understanding and control of the most basic processes of life herald revolutionary advance in biotechnology. In the few years since its introduction, CRISPR-Cas9 has been employed in labs all over the world. In the first 2 years, there were 2,000 published papers using this technology. It's generated thousands of publications and new experiments, and it's attracted over $1 billion in venture capital from private companies exploring its medical and commercial applications. The scope and versatility of this technology promises transformational impact as great as the discoveries of electricity, synthetic chemistry, and nuclear physics. If that sounds dramatic, just consider how life has changed with each of the discoveries, first in physics, then in chemistry in the 19th century and into the 20th century. Now the circle is being completed. We're gaining control, understanding of the final most complex dimensions of natural expressions, namely biology. And I think the revolution may be more subtle, but it's going to be truly dramatic. While much of this advance will be welcomed as constructive disruption, providing urgent and uncontroversial progress in biomedical science, agriculture, environmental and ecology, the power and depth of operation of these new tools in delivering previously unimagined possibilities for reworking or redeploying natural biological processes, some with startling and disquiet implications. All this should cause us to pay attention, think deeply, and seriously about what we're doing. So, for example, through alteration of sperm and egg precursor cells or direct action on early embryos, CRISPR opens the prospect of effective germline modifications. This creates an enormous range of opportunities for scientific studies, for improved animal crops, farm animals and for crops, and potentially heritable repair of human genetic diseases. But it also opens possibilities for large-scale manipulations and permanent alterations at the most basic levels of life, including human life. Together with advances in synthetic biology, CRISPR-Cas9 is being used to engineer complicated heritable biological circuits with improved or entirely new functions, including more efficient forms of bacterial biofuel production or new forms of photosynthesis, and possibly new programmable behaviors in animals, raising profound moral questions about our role and our rights within the order of nature. So, for example, scientists have created mice that glow in the dark. So, I mean, that didn't hurt them any, but you can imagine that we might be headed toward a whole array of sort of aesthetic, artistic projects. This is New York. You know what art does in New York. Would they not include this in their technology? And I think it's very possible. I've been saying this to my students for 25 years. I think we're going to eventually see a recombinant zoo of Dr. Seuss animals. I'm serious. There are really amazing possibilities here. So perhaps the most dramatic of all these and disquieting possibilities of this new technology are synthetic genetic modules known as CRISPR-based gene drives. They can simultaneously deliver both trait-altering genes and a mechanism that promotes their biological inheritance through a population. This creates a kind of mutagenic chain reaction, and it can alter or potentially eliminate an entire species, such as Zika-carrying mosquitoes. And this is being seriously considered to be done in Florida, right, Robin? Robin Cook is in the front row here. Robin's the author of all those famous medical novels, and he has a home in Florida. That's why it's okay, I'm pointing at you, Robin. All right. So this I think should be the subject of his next book, will be Zika-carrying mosquitoes and CRISPR. But it really, you can see right away, it raises profound questions. Do we know enough to do this? Are we going to upset whole ecosystems? Are we going to upset the foundations of human life? The challenges and opportunities of these new gene editing technologies are perhaps the most profound and difficult matters our species has ever encountered. Together with advances in cytology, systems biology, bioengineering, and the great advances in computer sciences that are making all this kind of stuff possible, these together open unprecedented powers for exploring and constructively deploying the fundamental forces of living nature in the service of human life, in the service of the good of the planet. Yet at the same time, they raise important questions about the appropriate projects and purposes for which they are employed. Beyond the prospects mentioned above, there are already proposals by serious and well-respected scientists for projects of broad ecological engineering, de-extinction of human species. That means bringing back ancestral human species like Neanderthals. We have the Neanderthal genome, by the way. We could perhaps do this. Also for bioengineering cures for aging, and by some calls, taking control of human evolution to advance ourselves to a post-human future. In a recent article, just last week, organisms created with synthetic DNA pathways for entirely new forms of life. This is where we're heading. This is amazing stuff. This article goes on to describe this as the starting point for efforts to create organisms with wholly unnatural attributes and traits not found elsewhere in nature. The cells constitute a stable form of semi-synthetic life and lay the foundations for achieving the central goal of synthetic biology, the creation of new life forms and functions. Well, good could come from that, but it sounds a little tricky, doesn't it? The question raised by such prospects go beyond issues of individual rights, of social responsibilities, to considerations of the very source and significance of the natural world, its integrated and interdependent processes, and the way these provide the foundational frame for the physical, psychological, and spiritual meaning of life. What I said in that complicated sentence is, we're going to have to be careful not to walk ourselves right off the stage of the drama of our deepest meaningful significance. In other words, we are made to fit into this world. Will we alter the world in a way where our lives have the same meaning and significance? The wise governance of these technologies is a key practical and moral challenge of our age, and our response to them could significantly alter the very future of life on Earth. As matters of basic human dignity, social justice, and responsible ecological stewardship, these issues transcend partisan politics and even national borders. I just gave a similar talk to universities in China, and serious issues. We're going to have to negotiate some very serious concerns here because there's a lot of money involved in this, a lot of commercial benefits, and, well, you can see where that's going. Anyway, serious stuff. So we're at the early edge of very challenging issues. And clearly these are species issues. They're not issues for any private group of people, elites or otherwise. They are issues for the whole human family, for the people who do the daily work of life, have the families, live and die. They're the people who need to decide this, not some people who place themselves as engineers over human life and human future. Individuals of the whole human family, with all the diversity and cultural perspectives of our species. Now at the early edge of this revolutionary revolution, as Huxley said, within the context of deepening public awareness and concern, we have an opportunity to consider these matters in an informed, comprehensive, and forward-looking way. And it's a very serious responsibility. Clearly, there are many considerations in the wise use of these powers, commercial, social, and legal, and all of these are extremely important. But as a physician, I want to speak with you tonight about their direct application in human life, in medical research and clinical care, and beyond therapy, in the pursuit of happiness and human perfection. So, we may be nearing the beginning of the end of genetic diseases. What a moment in the human story. For the first time in our long and troubled history, the tools appear to be in place for the conquest of diseases and disorders that have burdened our species since the dawn of time. This is Jennifer Doudna, a professor of molecular biology at UC Berkeley and a key figure in the discovery of CRISPR-Cas9. She made the statement. This little boy is evidence of the powerful potential of earlier and less efficient gene editing technologies, but he's a paradigm for what could come. He was cleared of his leukemia by genetically altered immune cells. But now with CRISPR-Cas9, with a revolution that's emerging, researchers are reporting dramatic gene editing advances in mouse models, so far only mouse models, in a wide range of genetic disorders, including sickle cell anemia, Duchenne's muscular dystrophy, and Fanconi's anemia. And 3 months ago, the Chinese announced that they had become the first group to treat a human patient, a person who had aggressive form of lung cancer, and they did this with cells that had been altered with CRISPR-Cas9 techniques. By the way, one of the big benefits of this is going to be very, very effective tools against cancer, very promising research right now. These therapies are done in adults or children, not in embryos at this stage. We'll get to that in a minute. These therapies are accomplished by removing circulating blood cells from the patient, some of the ones I listed anyway, altering them through gene editing and then putting them back into the bloodstream in ways that help the patient. This can also be done with a variety of cell types drawn from other tissues of the body, liver, pancreas, heart maybe. And then the cells are re-injected into the same organ or tissue. And for hard-to-reach or fragile organs, scientists have developed a range of clever ways to directly deliver the instruction-bearing CRISPR complexes using vehicles such as liposomes, little fat globules, or nanoparticles, or viruses that have been specially engineered to target specific tissues or organs of the body, including regions of the brain. You can see where this is going. We're going to get effective communication and alteration probably throughout the whole human body, targeting any specific region you want. The dramatic potential of such therapies is immediately obvious. For the first time, we will have power to treat hundreds, perhaps thousands of diseases and conditions, because how do you define a disease? We'll have a ability to intervene in many, many things, many of which are very serious. So, for example, there are at least 6,000 known single-gene diseases that afflict human beings. 95% of them have absolutely no treatment in the modern world as of now, and many of these, perhaps most or all of them, will become targets with CRISPR-Cas9. But it's obvious that in certain cases, the earlier the genetic intervention can be made, the better the treatment outcome will be in preventing the disease onset and allowing normal development. This has reawakened the long-held hope that we might one day be able to make targeted DNA modifications directly at the level of human genetics, at the gametes, at the sperm or egg, or in early embryos, and thereby remove a genetic disease once and for all from a family lineage. So this is a disease called Miller's syndrome. But here, this is one called Lesch-Nyhan's. And if just to highlight how significant this is. The people with this disease, the little children with this disease, actually have something that causes them to chew their own fingers off, and they chew their own lips off. It's very tragic. And they die usually in their late teens or early 20s. So very serious. But this in turn raises challenging questions about what degree of acceptable risk and what types of conditions should we count as worthy targets of our interventions. Are albinism and dyslexia conditions that we should treat? And what about parental preference for hair and eye color or taller stature? Red hair, for example, is controlled by a single gene. Most conditions we would care about, intelligence, beauty, longevity, are the result of interactions among many genes and would not be easy to target. But as we learn more about genetics and the genetic basis of depression, autism, Parkinson's disease, for example, we may be tempted to develop preemptive germline interventions. Harvard geneticist George Church has compiled a list of rare protective variants of large impact, which exist naturally in the human gene pool, but only in a few people. These include, here I'll show you the variants, these include stronger bones, leaner muscles, lower risk of coronary artery disease, cancer and diabetes, and greater resistance to viruses, all of which he believes might improve general health if they were just, engineered to be more common in the human genome. I was, by the way, I'll tell you more about this in the Q&A maybe, but I've started a project with Jennifer Doudna, the woman I showed earlier. She and I are organizing an international project to discuss and debate these issues. And we held our first meeting last weekend at Berkeley, and it was really an astonishing meeting, just absolutely cutting-edge science all the way. And George Church was there. He's a very interesting guy, but he's one of those guys where I had the feeling that, well, we want a guy like this in our species. He's like popcorn. He just comes up with ideas all over the place, and it's fantastic. He's wonderfully creative. But it might be also wise to have people who are, more historical and theological and philosophical perspectives to sort of contain all this energy and invention. Nonetheless, we need this conversation, and we need all parties in the conversation. So I really like George Church. I'd never talked with him at length before, and he's a very interesting, cool person to talk with. So George Church is pretty gung-ho on this and believes it's going to happen and believes it should happen. Another man at the meeting, very thoughtful developmental biologist, Stuart Newman, his response to such suggestions are different. Newman warns the genetic design of future offspring, even with a limited objective of making these future children more normal, will open the door to attempts to pick and choose other characteristics, because definitions of normality will vary, as will access to technology and willingness to take risks with future lives. Yet some argue that it's human nature and human destiny to improve ourselves, even at the most fundamental level of our genes. These include the transhumanists, an international intellectual and cultural movement advocating technologically mediated enhancement of human intellectual, physical, and psychological capacities. Their logo is H+, humans plus, improved human beings. I've had several of these members of the Transhumanist Society. In fact, my teaching assistant was one of the founders of it a few years ago. They're interesting, intellectual, and serious-minded students. They argue that our advancing technologies offer us the opportunity to escape the constraints and cruelties of an amoral evolutionary process, to lift humanity to the next level of personal and social flourishing as genetically enhanced human-machine hybrid post-humans. That's their terms. They start with the assumption that the world came from cruel and immoral. It's a coincidence within a chaos. So they're starting with a theological or it's a mystical assumption for where the world came from. Somehow they believe we do have, in spite of its terrible origins or whatever happened, coincidences, that somehow we did land with the wisdom to guide ourselves to the future, which is pretty interesting. So, on a less dramatic level, many point to the steady increase in medical interventions for purposes beyond therapy. The traditional role of medicine has been to cure disease and alleviate suffering, to restore the patient to a natural level of functioning and well-being. That's what most of us think of as medicine. The medical arts were in the service of a wider reverence and respect for the order of the created world. This idea was put succinctly by the Roman physician Galen when he said, "The physician is only nature's assistant." But now, with the powers of our advancing biotechnology, there's a new paradigm, one of liberation, technological transformation in the quest for happiness and human perfection. Grounded in the widespread practice and general acceptance of cosmetic surgery, slowly but steadily, the scope and purpose of medicine are being extended along the gradient of our appetites and our ambitions to encompass dimensions of life not previously considered matters of health, but natural human variations or limitations. From Rogaine, that's to cure baldness. Baldness was not a disease when I was trained at Stanford Medical School. So Rogaine, growth hormone for short stature, to birth control pills and Viagra, and more recently Provigil, a drug that allows prolonged periods of wakefulness up to 60 hours. You can really study well that way, I guess. And Seasonale, a technological bypass to the monthly periodicity of the natural menstrual cycle. In all these ways, we've altered and revised the given frame of nature, and we mostly have accepted these technological advances as a civilization. Increasingly, not only are we accepting these things, but increasingly we've come to expect from medicine not just freedom from disease, but freedom from distress, struggle, and even the constraints of a natural life process, from all that is unattractive, imperfect, or just inconvenient. We've developed the mindset. We're starting to think of human life as a technological problem to be dealt with by our scientific advances. Recognizing the extraordinary challenge and serious significance of these trends, the President's Council on Bioethics undertook a multi-year inquiry exploring the implications of employing our advancing biomedical technologies in the pursuit of the traditional realm, in projects beyond the traditional realm and reach of medicine. So in our report, I served on this committee during the Bush administration. In our report, Beyond Therapy, Biotechnology and the Pursuit of Happiness, we discuss the historical foundations of our quest for mastery over nature. It's not the long-term way we dealt with nature as peoples, but it's a modern, more Enlightenment perspective. We then went on to consider a series of specific applications of biotechnology beyond therapy that might one day be used not by radical reformers, but by ordinary people just wanting better lives for themselves and their children. That's where the temptation will be, you see. We discussed proposals ranging from better babies and superior performance to the quest for unaging bodies, projects that are likely to become targets of our advancing genome editing technologies. By the way, Google has they gave $1 billion to an organization, Calico, California Life Institute, and they're working for cures to aging. There's already an accepted social precedent for this kind of genetic alteration, in vitro fertilization, where people who want to get donor eggs seek out donors who are showing the best genes in their life expression of life. So this ran in the Stanford Daily. This was years ago, like 15 years ago, and one of the student I knew there at Stanford answered the ad. They ran this in all the Ivy League schools, Princeton, Yale, Harvard, those where the best genes end up. You know what I mean? So she was one of the two finalists, and of course they flew her back where the other one would be Harvard. And she said they put them up in a hotel room for a whole weekend and had them fill out 30 pages of detailed essay forms. They read them through, And then they invited her to meet the couple because she qualified. And in the meantime, I'd been on a television program. I'm going to waste a little more than 45 minutes, okay? It's more fun. I was invited on a television program. I was invited by every major media, and I didn't want to do it. But finally, I decided, okay, I'll do CNN. It was this thing called Talk Back Live. And there was an audience, and the other guest was a broker from the law firm who was mediating this deal. And somebody asked him, so why do they want such a tall, intelligent, athletic egg donor? So he had to be 5'10" and have 1,400 in the SATs. That was when the SATs were hard, by the way. So he said, well, it's easy. They're a tall, intelligent, athletic family. So that's what everybody was sort of expecting, I guess. She said that when she was finally introduced to the family, she couldn't believe it. The mother-to-be was 5 foot tall. And I suspect the father wanted a son and wanted the son to be tall. So they might have even been fertile. He didn't like her genes, maybe. I'm making that up, but I'm imagining why. But see, it's no longer science fiction. It's serious business. It's the tools, the powers are coming into our hands. Acknowledging these challenging prospects, there is within both the general public and the scientific community a growing apprehension that previous speculations regarding designer babies and even state-sponsored eugenics programs may now be, at least to some degree, technically feasible, and thus matters of serious practical concern. Moreover, there's worry that personal ambition, institutional competition, and even commercial interests, and I would add military interests, may lead to imprudent and premature human applications of these technologies, with potential clinical disasters and damage to the public trust. In early 2015, amid an accelerating pace of advance in gene editing technologies, a group of leading scientists, including Jennifer Doudna and several Nobel laureates, published a statement in the journal Science, citing what they called an urgent need for open discussion of the merits and risks of human genome modifications, and calling for an international discussion of these matters. Their central concern was the possibility that germline therapy might be attempted with human gametes or embryos before the difficult scientific and ethical issues associated with such procedures had been adequately considered. Later that year, together with the Chinese Academy of Sciences and the British Royal Society, the British Royal Academy, that is, the United States National Academy of Sciences hosted the International Summit on Human Gene Editing in Washington. So this was just 14 months ago. This meeting was made especially urgent by reports just a few months before by Chinese scientists of having used germline experiments, human embryos in germline experiments, albeit with non-viable embryos, so there was that. But everybody realized this is coming on. After 3 days of discussions, the organizing committee issued a statement recommending intensive basic and preclinical research, including, as necessary, studies using human embryos, but with the proviso that they should not be used to establish a pregnancy. They went on to declare that it would be, quote, irresponsible to proceed with any clinical use of germline editing until issues of safety, ethics, and social consensus had been adequately explored. I'm not sure how they're going to feel now about social consensus given our recent elections. But having said that, they called for it. So in making their recommendations for the way forward, it's clear that the organizing committee recognized the extraordinary opportunity. They knew there was great opportunity using gene editing tools for advances in biomedical science and potentially in clinical care. And most especially, they understood that these tools could illuminate the understanding of developmental biology. See, we spent the last century understanding the basic molecules. They're the ingredients, if you will, of how human beings are made. But now we want to see how the genes knit together to produce a living organism. So they know that developmental biology studies are key to the advances in the future. And so they want to encourage these studies. And in fact, prior to the official statement, several leading scientists, including some on the committee, had underscored the importance of these kind of studies. Nobel laureate, my teacher, in fact, at Stanford, Paul Berg, and David Baltimore had stated, these are necessary to address complex diseases like cancer. We must carry our investigations to the most fundamental elements of living systems. Likewise, George Daly, dean of Harvard Medical School, had affirmed gene editing could be used to engineer specific disease-related mutations in an embryo, which could then be used to produce embryonic stem cells that could act as models for testing drugs and other interventions for disease. Such studies with human embryos would extend 30 years of research done on mammalian embryogenesis using mouse models. But now we wanted to know what we could see with human studies. Nonetheless, it's important and it's notable that although the committee statement made prominent mention of ethical issues associated with the clinical application of germline therapies, that means for up and running human patients, it made no mention of ethical issues in the instrumental use of human embryos in scientific research. This is especially notable and surprising in light of the history of the worldwide controversies over embryonic cloning and embryonic stem cells. In certain nations with advanced research programs, such as Germany and the United States, these controversies have never been resolved, and policies that prohibit or constrain research on human embryos are still in place, and I'll tell you more about that in a minute. Although current U.S. laws and regulatory principles governing research on human embryos provide some limitations and constraints, these apply only to federally funded research. Many of our 50 states have no prohibitions, like California, for example, no prohibitions against embryo research, at least up to 14 days, when it's done with state and private funding. And several actively, like California, actively support such studies. However, since the NIH, that's the National Institutes of Health, you all know that, since the NIH provides the major source of research funding and the federal laws play a prominent role in this research, that's going to become a major controversy. And I predict, it's one of the reasons I want to talk to you about this, I predict that the controversies over the use of this research with human embryos is going to make the debates over embryonic stem cells look like mere prelude, just getting ready for the real large scale, and I want to explain to you why I think that. So other countries have different views of this, but you'd be surprised how many countries are conservative when it comes to embryo research. Major nations will not allow it. France, Italy, Germany will not allow you to create embryos and do research on them, and many other countries in the world. In the United Kingdom, however, embryos may be instrumentally used in research for up to 14 days of development. That's been the law so far. This bright line, beyond which one should not go, is based on a kind of received tradition that dates back to 1984 in the Warnock Commission, and they had very specific arguments for why 14 days was okay, up to 14 days was okay, but they did acknowledge that there is no particular part of the developmental process that is more important than any other. They just wanted to do the research, and they couldn't keep embryos alive more than about 9 days at that point, so they said 14 days. And now this principle is under reconsideration. I'll tell you more about this in a second, but there's now technological advances that allow us to keep embryos alive. We don't know how long, but they're now discussing whether they should keep them alive to 28 days. I'll get back to that. So 14 days will slip away. In the United States, the conflict over research using human embryos goes back several decades to controversies over federal funding of research to improve clinical techniques in vitro fertilization. After a long period of debate after in vitro fertilization came online, that was 1978 when that started, but the debate got going and went for a long time. After long debate, in 1995, the U.S. Congress passed the Dickey Amendment. Passed is not exactly the right word. It's a rider annually attached to the HHS budget. So this Dickey Amendment, here are the rules of it, it prohibits federal funding of research that involves the destruction or endangerment of human embryos or the creation of human embryos for purposes of research. This amendment, which has been renewed every year since, but is not encoded in law, reflects the idea that a large number of American citizens object to this research and to research on human embryos, and therefore should not have to effectively cooperate in that research by having their tax dollars funded. That was what the debate was all about 10 years ago. It was about federal funding. In the years following the Dickey Amendment and so forth, you all remember, with the cloning of Dolly the sheep, 1996, there was widespread public concern that cloning technology might soon be extended to interventions in human life. Such concerns were pretty much had to do with babies being born, but there were concerns about creating cloned embryos too. Such concerns were brought into focus when just 2 years later, human embryonic stem cells were purified, isolated from human embryos, left over from IVF clinics in this case. And then the suggestions came forward that if we could make tailor-made cloned embryos, we could create specifically tailored cell lines for harvesting to treat patients that wouldn't have immune response to them. You all probably remember that. Ron Reagan getting up at the National Convention and saying we need a toolbox or toolkit using cloned embryos. This controversy was magnified by the widespread belief that embryonic stem cells offered a shortcut and direct path to treatment of a wide range of diseases and disorders that affect millions of Americans. The figure was 143 million Americans could benefit from these treatments. And there was reason to think that if the therapies couldn't come from the cells themselves, we would learn enough with the cells to gain control over these crucial developmental processes. The controversy quickly became characterized as one of religious objection versus scientific promise. At my own medical school, you remember this, it became a kind of stem cell wars, all the promise. We just start with a basic cell type and learn to produce all the different cells, inject them into the heart and the liver and cure everybody. In my own medical school, the Stanford Alumni magazine had a cover story that perpetuated this false dichotomy of religion versus science. In fact, many non-religious people also objected to embryo destructive research on the grounds that it involved a basic violation of human rights. And it was a very intense debate. You remember, perhaps, that President Bush's first address to the nation on August 9th, 2001, was on the issue of stem cell debate. It was on the issue of stem cell research, and it was during that debate that he announced the formation of the President's Council on Bioethics. And so this was a very hot issue in our society, but then a month later, 9/11 happened, and that changed the landscape quite a bit. So this went on, and I had a front seat on these controversies being on the council. I want to show you a clip of a political ad that ran in 2006, and it's a window into how deep and difficult this controversy can become, because it's portrayed as science versus suffering. What happened there? So the culture wars. Here we go again. Temporarily, the development of so-called skin cells to stem cells solved it for the moment, and pretty good tool. Yamanaka from China got the Nobel Prize for that. And then with President Obama taking office, he ruled that more lines could be created, and it's been on hold, back burner sort of stuff since then. Now with the change of administration and the new advances in science, controversies may reemerge, some arguing that embryo research is wrong in principle, others arguing that the promise of scientific knowledge and therapeutic advance make it a moral imperative. This is a political ad, pro-embryo research, but I thought it was ironic. We fight because lives can't wait, but the pro-life community says, but their lives are being destroyed in the process. So This is an 8-cell embryo. There is no statement of why our country takes the position it does, but it's fundamentally based on the notion that human life is a continuum for fertilization on through all its developmental stages, which go on till death. This is an 8-cell embryo on the sharp tip of a pin. That's how tiny it is, but it is the beginning of a human life. And every one of us in this room started out like that and went through the stages of life development until we were born and then continued to develop onward. So it's a very, very tense and difficult debate. So I want to just take a few minutes and tell you where I think this is going and show you why I think this is going to be a very difficult transition in our civilization. There's an old Chinese saying, those who choose the beginnings of a road also choose its destination. So because of this new opportunity, we need to, and because of these issues were never resolved, we need to ask ourselves 4 basic questions. Will we now endorse the use of human embryos for a wide range of studies of infertility and early development? There's a whole range of things we could do, including knocking genes out, seeing what they did, knocking new forms of genes in, studying embryos subject to industrial chemicals to see what damage they do, studying making disease models. This is Rudolf Janisch from MIT. Rudy was at our meeting last weekend too. And he's the first guy to develop mouse models of recombinant mice. And he's a very astonishing scientist. And now he's told us at our meeting that whereas it took him 2 years to make a mouse model, now he can make one for very cheap within a few months. And so this is going to be a huge temptation, what we're going to do. Because a lot of good science could be learned this way, but at what cost? It's very important to realize that certain countries are already given the go-ahead for this. UK is already doing, or preparing to do anyway, research on human embryos, knocking genes in, knocking them out. They're doing it in China. Last week a guy told me in many places in China, and they're doing it in Sweden as well. This is the researcher in the UK who's doing it. And I do not doubt that good science could be learned that way. The question is it the proper cost? The next question, will we allow the creation of embryos specifically for research purposes? And here the issue comes up that there are certain kinds of things you just can't do you can't learn without being involved in the creation of embryos. And likewise, you could create cloned embryos because then you'd have standardized batches of embryos that you could do different alterations and see how they differed in their effects. And so this brings us to the third question, how many embryos it is okay to use for research? And here you can see how, especially if you're going to use cloned embryos, making many identical copies, like this Japanese scientist made 581 identical copies of mouse embryos, and he could do very interesting studies with those similar models. So the bottleneck for that, of course, is human eggs, and human eggs are in rare supply. You have to talk somebody into being super ovulated, and then they only get 8 or 10 or 12 eggs out of it, and it costs a lot medically and in terms of discomfort for a woman, but now there's suggestions that we might be able to get eggs from aborted fetuses because they have many more eggs than adults are lost across the span of development. And furthermore, it was suggested that, and now clinical scientific studies with mice show that looks like we're going to be able to make eggs and sperm out of embryonic type stem cells, pluripotent stem cells. So this is, of course, the sort of the most promising opportunity from the standpoint of this agenda, because you could have millions of gametes then. You wouldn't be limited by the number of women willing to donate their eggs at some medical risk and great cost. And I had a conversation with this guy, Davos Salter. This is quoted in Nature magazine about 10 years ago. He saw it coming. You could have millions of gametes, eggs and sperm. We already have plenty of sperm, you know that. That's readily available. But it's the eggs that are the problem, okay? You could have millions of gametes that could be combined at will. Today, you can't experiment on human embryos because it's considered morally repugnant. and they are difficult to get. If embryos could be grown in culture like any other cell, this latter problem would disappear. They'd become like any other cell type. They would become objects and would be used as objects. I have no idea what kind of moral value or rights we would give to those embryos. We'll probably go through the same agonizing we did with IVF. It could be terrible to begin with, but then it'll become a fact of life. Ironic word there. Maybe 20, 30 years from now, we'll read in the newspaper that someone made 20,000 embryos and studied their development, and we'll decide it's okay. So that brings us to the fourth and most challenging question. How long would we keep such embryos alive if we were to do it? Because up till now, no country has allowed to go beyond 14 days, officially anyway. So it's obvious if scientific knowledge of human development or useful models of disease can be created through gene editing of early embryos, then the study of embryos beyond 14 days would provide valuable additional information. With laboratory studies of embryogenesis beyond 14 days, the entire process of human organ development would be studyable, okay? Moreover, it's increasingly clear that progress with pluripotent stem cells, which even if you had standardized batches, it's very hard to coax them up to learn about how to do them. So the more the better, and the longer the better. It's increasingly clear, moreover, that, and that's why the UK is interested in going beyond, but it's also increasingly clear that all those promised therapies haven't come about because it's very hard to coax these cells. But the place where the cells do get coaxed to be just the right kind of cells that make cells, tissues, and organs is within the organism itself. So you'd want to be able to both study it and maybe even use the products that were formed by developing embryos. Accordingly, there are now serious scientific and commercial efforts to obtain useful products from human embryos and fetuses. In fact, in many stem cell research reports showing therapeutic promise, the stem cells used in the trials are actually taken from aborted fetuses. Adult stem cells are anything after the embryonic stage for 5 or 6 days. All the rest are called adult stem cells. That's why we're all hearing about adult stem cell research, but not realizing that they're taken from aborted fetuses. So Sir Richard Gardner, a very leading developmental biologist at the University of Oxford, suggested that kidneys and livers from aborted fetuses could be given to the desperately ill and thereby ease the shortage of donor organs. He goes on to say that fetal tissues may offer a more realistic solution to the lack of organs than other technologies being developed, that he was surprised the possibility had not been considered, and that experiments in mice have shown that fetal kidneys grow extremely quickly when transplanted to adult animals. Moreover, it was recently reported that a Silicon Valley biotechnology company is specifically dedicated to developing and facilitating obtaining fetal organs from abortions and making them clinically acceptable. I don't know how well they're doing on that. At least one bioethicist suggested that cloned human embryos should be created, implanted, gestated, and harvested for the production of medically useful cells, tissues, and organs. Although there are to date no reports of successful implantation and gestation of any primate clone, including human beings, I know the guy who cloned the first human embryo, and he says he could do it if he just had more eggs to work with, and I believe him. He's a really good scientist. Good, at least in the scientific sense. I don't agree with some of what he does, but he's also the guy who developed the 3-parent embryo, by the way. But in spite of the fact, there are still advocates for this because they think it's coming. Oxford physician and professor of philosophy Julian Salvalescu writes, "It is morally required that we employ cloning to produce embryos or fetuses for the sake of providing cells, tissues, and even organs for therapy, followed by abortion of the embryo or fetus." During the embryonic stem cell debates, arguments were already being made in favor of research beyond 14 days. A variety of criteria were proposed for the moral boundary of inviolability, with some suggesting sentience, feeling things, others awareness of pain, and some even saying conscious self-awareness should be the criterion. Associate Professor of Neonatal Medicine and Bioethics, Dominic Wilkinson, also of the University of Oxford, states, "It may be that research using early human embryos is ethically preferable to research in animals, since it would not cause the research subject to suffer, and research findings are more likely to be directly transferable to humans." And Princeton philosopher Elizabeth Harman has written, "In my view, fetuses that die before they're ever conscious really are a lot like plants. They're living things, but there's nothing about them that would make us think that they count morally in the way that people do." My Stanford colleague, Hank Greeley, who served on the California Cloning Commission, which recommended embryo research through the 14th day, is quoted as saying, "We did not mean that 14 days would always be the limit." He was challenged at a conference I was at. I heard him say this, only he said it more bluntly and harshly. We did not mean that 14 days would always be the limit. That could be changed in the future based on understandings that would likely come from neuroscience. Well, what understanding that we get from neuroscience would allow us to keep embryos alive longer than the previous criteria? Hank's a very nice guy. I like him very much, but I don't agree with him about this stuff. Another colleague, Ernle Young at Stanford, who started the Bioethics Center, a Methodist minister, by the way, he wrote, "The blastocyst or embryo is a human organism, but not a person. It does not even become a potential person until the emergence of a rudimentary central nervous system, something which is thought to happen at about 26 weeks of gestation. Therefore," he goes on, quoting a legal scholar, "since embryos do not have the requisite biological structures to generate and sustain the mental capacities, embryos cannot be benefited or harmed. Therefore, what you do with them shouldn't be a moral issue." 26 weeks is 2/3 of the way through natural gestation, and babies born prematurely at that age are routinely cared for and generally go on to live full and happy lives. I debated Julian Salvalescu, the guy I quoted a few minutes ago about how we should clone and gestate and harvest. I debated Julian Salvalescu here in New York at a public forum. And I asked him how far into development he would allow these clone fetuses to grow and develop before they would be harvested for transplantable parts. He replied that he would find 6 months, 26 weeks, morally acceptable. Some years ago, in an earlier era of optimism about germline gene therapy, the prominent geneticist French Anderson suggested that women who intend to abort a fetus with a genetic disease should volunteer for gene therapy trials, then delay their abortion to allow time to see if the therapy worked. More recently, some have raised the disturbing but strangely logical suggestion that since embryos left over from IVF are being used to obtain embryonic stem cells, why not use them for projects that include their genetic alteration, implantation, and later extraction of their cells, tissues, and organs? This is just an extension of the argument that says they were going to die anyway, so why not gain something good from their life? Well, it's a strange moment in human civilization. Here's a quote from from Columbia University, I believe. Erwin Chargaff made major advances in genetics in his distinguished career. And he warned us, "Science is now the craft of the manipulation, substitution, and deflection of the forces of nature. What I see coming is a gigantic slaughterhouse, an Auschwitz, in which valuable enzymes, hormones, and so on will be extracted instead of gold teeth." Of course, there is historical precedent for all this. It's been reported, I don't want to show you that slide yet. It's been reported that Francis Crick, discovered the double helix, he got the Nobel Prize. It's reported that he said no newborn infant should be declared human until it has passed certain tests regarding its genetic endowment, and that if it fails these tests, it forfeits the right to live. His co-discoverer, also Nobel laureate, James Watson, has suggested that the offspring of those with the lowest intelligence be genetically engineered for improved intellectual capacities. And when there are mistakes in these scientific projects, and there are children born defective as a result of science's attempts to manipulate life, he has urged that doctors attending the birth of laboratory-conceived human babies be given the right to terminate the lives of the infants when their mistakes are made, when it didn't come out right. The French philosopher Voltaire once said, "What is madness? To have erroneous perceptions and to reason correctly from them." The American physician, Leo Alexander, was present at the testimony of the Nazi doctors at the Nuremberg trials and later authored the Nuremberg Code. In his compelling essay, Medicine Under Dictatorship, he explains how the Nazi program of racial hygiene, an attempt to improve the human species through genetic selection, became one of the worst horrors of modern history. He wrote in this essay, "Whatever proportions these crimes finally assumed, it became evident to all who investigated them that they had started from small beginnings. The beginnings at first were merely a subtle shift in emphasis in the basic attitude of the physicians. It started with the acceptance of the attitude basic in the euthanasia movement, that there is such a thing as life not worthy to be lived." Now, as we gain increasing control over the very molecular basis of life, we need to pause to consider carefully how we should proceed. Within the scientific community and the general public, there is wide recognition that our new gene editing tools are a threshold technology, an opportunity for us to re-envision and reorder our place and purpose within the natural world. The issues involved go beyond biology. Our whole sense of personal and social destiny may hang in the balance. There is another old saying, "The first principle of intelligent tinkering is to not throw away any of the parts." We must now not be so foolish as to arrogantly ignore the natural foundations that are the very frame of our human freedom, nor the treasures of our social, philosophical, and spiritual traditions that make that freedom truly free. The very future of global civilization may depend on this. Thank you.